Good morning. Thought it was about time for another tea and tingles. I had some genetic stuff that I talked about in one, about inheritance, Mendelian genetics, and I thought I would do one today on what genes, what DNA actually does. So I figure that's maybe not something that most people know. Today's tea is a raspberry black tea. It's a Ceylon tea with some dried raspberries in it. It also has blackberry leaves, rose petals, and natural flavors. Which one of those? One of these days I'm going to ask them what natural flavors they are, but that's what it says. I think I have my scoop up here. Doesn't look it. Oh, let me show you. Today I actually brought my tea ball. It's mesh. It has a little hinge here that clips it together. And it also has this little sunflower charm on it, which is made out of clay or something, heavy enough to hold it so it doesn't slide into your cup. You can see I've shortened it a bit here. This is what the tea looks like. You can see the black tea there, and these are the rose petals. And this right here, that's a little dried raspberry. It smells like raspberries. Very fruity scent. If you have raspberries or blackberries in your backyard, in the summertime, when raspberries are out, which is late August here, for about 2 weeks we have raspberries. This is what it smells like when you're in the raspberry patch. When I was very small, not very small, 5 or 6, I had a neighbor who was about my age, and they had a huge raspberry patch behind their house. We used to have a place, we had a wooden fence between our backyards, not up at the front where the driveways were, but toward the back. But you could sneak around the back of the wooden fence into his yard. That was our secret passage. There were some cedar trees that had been planted along a cyclone fence, a metal fence, which was at the back part of the property to separate us from the guy who lived on the other side, who was an older man, who we didn't talk to much, but he mowed his lawn all the time. A huge lawn, and he mowed it all the time. I think that was his purpose in life. Anyway, so we would sneak around through the cedar bushes, cedar shrubs, and around the back of that fence. And then if you went down to the right, along the edge of the property, you could be very sneaky. And you could duck under the raspberry bushes. And there was like a little cave of raspberry bushes. And we'd sit in the middle and pluck raspberries off the branches and eat them. It was pretty magical for a 6-year-old. They probably weren't nearly as big as I remember them, but they seemed huge. This is the cup I'm drinking out of today. You can hear it's just a ceramic. This one that has sort of a matte finish to it. But then this little glossy streak. I have 3 cups like this in different colors At least one of which has a crack in it, so I can't use it as a teacup anymore. But this one I still have. And it makes a nice sound. Alright, so here's my tea. Alright, so that will take about 5 minutes to steep. Okay, I'm looking at my watch like I'm going to remember. I have no idea. Okay, so I actually started this video a week or so ago when I was visiting my brother-in-law, and then I ran out of time. So I'm a little unsure of what I started with and where to go over things. Just rambling now. I have some sparkly pipe cleaners that I'm going to show you. I know you're probably wondering why I have these. I was going to use these when I was talking about inheritance in the other video, but I didn't. And now what I'm going to do is show you a little bit about DNA with these. I think it will work. We'll see. So, when I talked about inheritance before, Mendelian genetics, actually I think I have it here. Biology textbook. I'd show it to you, but there are probably better ones now. And I want that tea so much right now. Okay. So we talked about Mendelian genetics. A monk named Mendel who observed pea plants because he had nothing better to do. And came up with this idea, came up with some laws of genetics, one of which being inheritance, that there was some trait that was common to the parent that got passed on to the offspring. So the example we used in that one was about flower color, purple or white flowers. And we also talked about dominant genes versus recessive genes. So dominant genes are the genes that are expressed that will overshadow any recessive genes. But if you have two copies of a recessive gene, then you'll see that expression. You'll see that trait. In this case, if you have one purple gene, regardless of whether or not you have a white gene, you also have a purple flower. So white is the recessive gene. The only way you have white flowers is to have two white genes. We talked a little bit about that being homozygous, when they have two of the same copy. So if they have two purple genes, then they're homozygous. If they have two white genes, they're homozygous. But if they have one of each, then they're heterozygous. Homo meaning the same, hetero meaning different. And then we also talked about how heterozygous heterozygous genotypes, we call that a heterozygous genotype, how that hides genes in your population. So if you cross purple and white, you have one generation of entirely heterozygous flowers, one purple, one white gene. And then if you cross these flowers, you end up with purple and white flowers, some homozygous and some heterozygous. And in fact, that's in the proportion 3-1. 3 of the dominant trait to 1 of the recessive trait. That's if you have heterozygous parents. And actually, as a side note, I won't do the math for you, but if you cross, this is one of the ways of doing simple genetic testing for traits that follow Mendelian genetics, is to cross strains and then look at the proportion of the offspring that have these traits. So if you had one that was a dominant purple and a dominant in a heterozygous purple, and you crossed them, then you would have just purple offspring again. So you'd know that they were not both heterozygous. You could do it in various combinations until you figure out which ones are which. Okay. So, what I thought I'd talk about today is, well, what does that really mean in terms of what genetics does? Hang on. Tea's ready. After that brief review, which I could have just said, go watch the other video if you want to know that first, but I didn't. Okay, put that down. Man, that's good tea. Just a little bit sweet. Okay, so we talked about inheritance, and you might have learned about, eye color, for instance. That's a popular example to use in high school biology when talking about genetics. Eye color is actually a lot more complicated than that, but we'll call it good. And there are multi-allele, so that with flower color we talked about just purple and white. Alleles being the different types of the gene, that are the different characteristics that are possible, either purple or white flavors of a gene is an allele. And there are ones that have more than just two. Eye color is one of those. That's why it's more complicated than all that. Depends on where the mutation in the gene is. But that's what I'm getting to right now. So what does all that mean? Talking about genes as coding for components of your body or of any organism. And what that means is, so you have DNA in your body. DNA is deoxyribonucleic acid. Nucleic meaning it's in the nucleus of a cell, nucleic acid. Deoxyribose, that part of it, describes the molecular configuration of the backbone of your genetic material. There's not going to be a test on it, so don't worry. But it's twisted in a helix in your body. I don't know if you can see that. So if these are, it has two backbones, two strands that are twisted in a helix in your body. And between the strands, there are base pairs. I'm going to write it down. They match each other. So there are four. That's it. Four bases, and they go into two pairs. So you have adenosine, I think. Tyrosine, T. I think I'm getting these names right. I'm not sure. I didn't look it up beforehand. Apologize. I can tell you it's A and T. Then guanine and cytosine, I think, are the other two. C and G. So, the thing about the base pairs is they only go with each other. They fit together like puzzle pieces. So wherever there's an A on one strand, there's always a T on the other. And the same with C and G. You don't have T and C go together, and you don't have G and A go together. That doesn't happen. Generally speaking, there's a whole thing that I could do. Maybe I'll do another video about that, about how mutations happen, and how genetic issues, bunches of genetic issues. But this is the way it's designed. That T and A fit together. The way these are just molecules, by the way. So the way the molecules are shaped, These two fit together, and these two fit together. Okay. So what happens is you have a sequence of these on that make up this genetic strand, right? So a gene is actually a sequence of base pairs, and I'm just going to make one up. Okay. This is one side. Gene actually is only one part, one half of your DNA. There could be a different gene on the other side. So it's a sequence of these base pairs. And I mean, you have millions of these in your body, in your genome, in your cells. And so this forms a code. And as a matter of fact, 3 of these, they go by 3s. I'm going to break that up here. 3 are a codon. They're called a codon. Just like that. Like codon, but without the E, because it's not codon. Codon. And a codon, what a codon codes for, in the language of your genes, is 1 amino acid. So there are 4 bases and 3 positions. So if you do the math, it's 4 times 4 times 4 possible combinations of base pairs. So there are actually 64 codons that are possible. But there are only actually, I think, 22 amino acids. So some codons actually code for the same amino acid. They're actually, I have a table. A few, maybe one, amino acid has only one code, but most amino acids have more than one. Let's see if this has a table in it. Amino groups, amino acids, here we go. Let's try triplet codes for, let's try that. There we go, this looks right. So this table, can you see it? Yep. Shows all the possible combinations of base pairs. And then what amino acid they code for. So I'm looking at this, and leucine, for instance, has 6, looks like 6 different combinations. And but tryptophan, which some people know from turkey, makes you sleepy because it's a serotonin precursor. Don't know if you knew that. Only has 1. There are some that have 2, aspartic acid and glutamate. Each have 2. Actually, a bunch of these have 2. Point is that they're there. Now, when I showed you that table, you may have noticed that it had U on it instead of T. So here's a thing that happens. when your body's ready to create this amino acid chain, which by the way, it's a polypeptide, but basically it's a protein, is what it synthesizes. That's what each of these amino acids are, building blocks for proteins in your body. Very cool. So it puts them together in a particular order, and there you go. Ta-da, you have some awesome thing that your body needed. But what happens is it separates the DNA chain, and then it has a little molecule that sits on it, and it starts at wherever the start codon is, because there's actually a code for start here. Because if you look at this and you have a big long sequence of letters, if you don't have a start codon, you don't know where the triplets start. So it could be, call this a reading frame. If you started one base off of what I showed you here, you'd actually have these 3 codons instead. Or they could go here. So you actually have 3 possible reading frames on this sequence that I just put up. Could be these 3, could be these 3, like this. So it has a start, and they have a start and stop codons. And it's a little bit more complicated than that too, because you have to have, in order for the molecule to fit down on there correctly, you have to have the right, they call it upstream, you have to have the right sequence further up before where you start reading the DNA up. I don't want to confuse you too much. Anyway, so they separate the strands, the molecule sits on here, and it reads off this DNA sequence, and it reads it off, it makes a copy of it. It's called transcription. But it doesn't make a copy of it in DNA. It makes a copy of it as RNA. RNA is just ribonucleic acid, so instead of the deoxy, it's ribo. And because it has a different backbone, it has a different base pair that matches with the A. It's U. Urine? Man, I don't remember. But it's a U instead of a T when it's RNA. It's one of the ways you can tell it's RNA. Anyway, so it makes a copy of it, writes it down on a piece of paper, basically. It says, okay, this is the sequence that you need. And then that gets handed off to another molecule that then takes, they grab amino acids and they stick them on in order. And then what you've got is this nice protein by the time you're done. It's very cool. And so what does that mean is, I'm getting to the point, right? When you have, for instance, purple, a gene for purple, then what happens is you read off purple, it creates a purple molecule, a purple protein. If you have a gene for white, it reads off white. But white is a different genetic sequence than purple. So where you might have, and of course genes are a lot longer than this, many, many more base pairs than this, but I'm not going to write out a 64K base sequence. We'll have to use our imaginations. So let's say that this is your sequence for purple. White would be maybe a simply different, what we would call a single point mutation. Which I've done here, right here. Circle it so you can see it a little better. Put a box around it. Let's say this is purple here. This is white. Okay. So it would be a single point mutation. They would call this a single base pair that's different. So maybe you have this G in purple, the T in the white molecule, and that is enough to make the protein a different shape, so it's a different color. Who knows? Sometimes, I don't want to get too much into it if I'm going to do another video over it, but sometimes, and this is what happens with eye color, is that mutation actually causes a stop codon halfway through the molecule. So let's say if CTT were the stop codon, which I don't think it is. But I don't remember. But let's say it had the single point mutation, right? But that meant stop instead. So you'd end up with purple having these 4 lovely 4 lovely amino acids making its purple color. It's going to look kind of like a sausage, right? But the white one would only have 1. Like that. And it would ignore the rest of this, because this says stop. So that's what actually happens with eye color. If I remember correctly, I might not be 100% on this, but brown pigment is a long polypeptide, a long protein chain. But blue eyes are actually a truncated version of that. It splits about, I don't know, halfway through or something. So you actually end up with a blue color instead of a brown color in the pigment. It's the same with green eyes are, I think, a little bit, I want to say they're a little bit longer, but they're less common because it's a mutation at a different point, which is interesting. Like I said, I'm not 100% sure on that. I'm only kind of sure on that. A little bit. Fairly sure. Anyway. So hopefully I haven't lost you horribly along the way. I just wanted to give you an idea of when we talk about genetics, what are we actually talking about, you know? When we talk about gene therapy, what is it that we're fixing? And what we're fixing is this genetic code that's defective in some way, or it's creating proteins that aren't beneficial to our body, or it's not creating enough protein. There are a lot of different ways to fix that stuff because there's a lot of moving parts. But when we're talking about inheriting genes, what we're talking about is inheriting specific sequences for coding different proteins which make up the components of our bodies. Kind of neat. At least I hope you think so. Anyway, that's all for now. Let me know if you like this video, if you want to hear some more about different weird things about genetics. Okay, they're not really weird, but just different things about genetics. Or if there's some other topic that I might know something about that you would like to hear about. I hope you have a good day.